Hello everyone, so in this video we are going to discuss about the MCQs related to protein synthesis. Okay, so very first question is which organelle is primarily responsible for protein synthesis. So basically this question is asking about the site of synthesis of protein. So ribosome is a site or you can say is a factory site for the protein synthesis. So right answer is option B. Ribosome are the site for where amino acids are assembled into protein based on mRNA instruction. Question number 2. What is the process of synthesizing mRNA from DNA? So the formation of mRNA from DNA is known as transcription. So basically you are transcripting the information from DNA in the form of mRNA. So transcription is a process where DNA is used to make a complementary mRNA strand. Okay. Question number 3. Which RNA carry amino acid to the ribosome? So generally transfer RNA is responsible for carrying amino acid to the site of protein synthesis, that is ribosome. So tRNA carry specific amino acid to the ribosome during protein synthesis. Question number 4. What is the start codon for protein synthesis in most of the organisms? So start codon in most of the organisms is AUG, that is that code for methionine. Methionine is a type of amino acid. Okay, so whenever there is protein synthesis taking place, it will start from the codon on mRNA, that is AUG. Okay, and signal the beginning of translation. Okay, now come to the question number 5. Which type of RNA make the majority of ribosome? So its answer is rRNA, because rRNA, that is ribosomal RNA, form the structure and enzymatic part of ribosome. Now come to the question number 6. In eukaryotes, transcription occur in. So what are eukaryotes? Eukaryotes have a well-defined nuclear membrane. Okay, and genetic material is present inside the nucleus. Okay, so eukaryotes transcription occur in mRNA is synthesized in nucleus and then transported to cytoplasm for translation. Okay, so transcription generally take place in the nucleus, that is option C is the right answer. Okay, and translation take place in the cytoplasm of the cell, that is protein synthesis take place in the cytoplasm of the cell, because ribosome is present in the cytoplasm of the cell. Question number 7. What enzyme synthesize RNA from DNA? So answer is option number B, that is RNA polymerase, because RNA polymerase read the DNA and make RNA copy. Question number 8. What happened during translation? So during translation it is a process of decoding mRNA. mRNA क्या करता है, प्रोटीन सिंथेसिस की इन्फॉर्मेशन डीएनए से लेके आता है और mRNA के ऊपर कोड के फॉर्म में ये इन्फॉर्मेशन प्रेजेंट रहती है. तो अब इन कोड को डिकोड किया जाता है राइबोसोमल साइट पर. ओके, सो ट्रांसलेशन क्या होता है? इट इज़ डिकोडिंग ऑफ mRNA इनटू प्रोटीन सीक्वेंस. सो प्रोटीन इज सिंथेसाइज हियर. ओके, ट्रांसलेशन में क्या होता है? प्रोटीन सिंथेसिस होता है. क्वेश्चन नंबर 9. हाउ मेनी न्यूक्लियोटाइड्स आर न्यूक्लियोटाइड form a codon? So generally codon is made up of is a combination of 3 nucleotides. Okay, so its answer is option number C. Codon is a triplet of nucleotide and mRNA that code on mRNA did code for one amino acid. Okay. Question number 10. Anticodon is found on. So anticodon are found on tRNA. Okay, and anticodon on tRNA are complementary to mRNA codon. Question number 11. What stop translation process? So translation start from start codon and stop at stop codon. Okay, so there are 3 stop codon are there. Universal, they are universal stop codon, that is UAA, UAG and UGA are the stop codon that signals the end of translation. Question number 12. Which molecule provide energy during protein synthesis? So during protein synthesis, ATP and also GTP provide energy for bond formation and ribosome movement. Question number 13. Which site of on ribosome hold the growing polypeptide chain? So it is the P site. Okay, that is peptidyl site hold tRNA carrying the growing peptide chain. Question number 14, peptide bond are formed by. So generally peptide bond are formed by between the amino acids. Okay, so we already know that the protein are polymer of amino acids, and these amino acids are held together by a peptide bond. Okay, question number 15 is splicing of intron occur in. So splicing in eukaryotes, intron are removed from mRNA in the nucleus. Okay, so nucleus is the right answer. Splicing of intron occur in nucleus. Question number 16, which of the following is not involved in translation? So in translation, DNA is not directly involved in translation. mRNA is involved, rRNA is involved, tRNA is involved, but DNA is not directly involved. Question number 17, the direction of mRNA translation is. So the direction of mRNA, ribosome read mRNA from 5' to 3' end. Okay, so option A is correct. Question number 18, what does mRNA stand for? So mRNA stand for messenger RNA. Okay, because mRNA carries the genetic information from DNA to the ribosome, genetic code. Okay, now question number 19, in prokaryote transcription and translation are. So in prokaryotes like bacteria, both the process occurs simultaneously in the cytoplasm of the cell. Okay, so option it is a coupled process. So option B is the correct answer. Question number 20, the first amino acid in any newly made polypeptide is. So very first amino acid that is present on newly made protein is methionine, because the start codon, protein synthesis start with the start codon AUG, and AUG code for methionine. So first amino acid is methionine. Question number 21, what is the function of promoter in transcription? So the promoter are DNA sequence. Promoters क्या होते हैं? DNA sequence होते हैं where RNA polymerase bind to start transcription. Okay, so it is there to start the transcription process. Question number 22, which molecule act as a adapter in protein synthesis? So which molecule act as a adapter in protein synthesis? So tRNA adapt the mRNA codon to correct the amino acid sequence. Okay, that is tRNA is the right answer. Option B is correct. Question number 23, what is polysome? Okay, so the polysome is multiple ribosome translating a single mRNA strand from a polysome. Okay, so polysome is mRNA sequence, a group of ribosome on mRNA. Question number 24, post-translational modification include all except. So post translation के बाद क्या modification include होते हैं? Except. So there is splicing occur before translation. Splicing जो है वो translation के पहले होता है, but phosphorylation is there after translation, glycosylation is there, methylation is there, and other happen after the protein is made. Question number 25, which of the following antibiotic inhibit protein synthesis? So the right answer is tetracycline blocks the site A of the ribosome in bacteria and retard the process of protein synthesis and inhibit the process of protein synthesis.